Welcome everyone to this panel discussion on disease-modifying therapies for Parkinson's disease. We have a great panel of speakers with us here today, and you will all be able to ask your questions. Don't be afraid to ask the hard questions. And the way you can do this is by using the app, the ADPD 2017 app that you can download onto your phone, and then go to the forum session, and there's an icon where you can ask your questions. You just click on it. So please feel free to ask questions. We definitely want everybody to have an opportunity to ask these guys the hard questions. So the topic, as I said, is disease-modifying therapies for Parkinson's disease. And many years ago, when I was a graduate student, I naively thought that Parkinson's disease was going to be easy. It seems so simple. You protect the dopamine cells, you save the dopamine cells. One small problem, the motor symptoms go away, and the disease is cured. Well, of course, it's proved to be not quite as simple as I thought. And many years later, we still aren't close to a cure. We don't have therapies that change the course of the disease or stop the disease. And we need to hold these guys to task for working on this and not getting to the finish line yet. So I want to give each of the speakers just a couple minutes to introduce themselves and maybe say a word or two about their work on disease-modifying therapies, and then we'll open it up and have you ask your questions. Okay, so I'm just going to go down the line and let each of you introduce yourselves briefly. Eliezer Mazga, we'll start with you.
Hi, good afternoon. Thank you very much for inviting me to be here, and thank you for you to be here today. For the past 30 years, I've been a professor at the University of California, San Diego Neurosciences Department, working on Alzheimer's and Parkinson's disease, developing animal models and therapeutics for the disease. We were lucky to collaborate with our colleagues in Prothena, developing immunotherapy approaches for Alzheimer's disease, and also small molecules and other approaches for the disease, targeting primarily alpha-synuclein. And more recently, about 8 months ago, I moved from San Diego to Bethesda, where I'm now director of the Division of Neurosciences at the National Institutes on Aging, where primarily advancing the national plan to address Alzheimer's disease, collaborating with Walter and our colleagues at the NIH toward the 2025 goal of finding a cure for Alzheimer's, but also for related dementias like dementia with Lewy body, frontotemporal, and vascular disease. Thank you.
And Peter Lansbury?
Hi, I'm a professor of neurology at Harvard Medical School, currently on leave, and chief scientific officer of a company called Lysosomal Therapeutics. We're working on an allosteric activator for the enzyme glucocerebrosidase, which many of you hopefully have heard of. The rationale behind this is very simple. There are many mutations and polymorphisms present in the PD population, which cause a loss in function in glucocerebrosidase. They also cause an increased risk of Parkinson's disease, and in patients who have Parkinson's disease, they cause an increased rate of progression of the disease. And in fact, the degree of functional loss in the enzyme is correlated to the increased rate of progression. So the idea is very simple. We're going to restore the normal activity of the enzyme, and we're going to do it with a small molecule. We have an allosteric activator that goes into man in mid-year for safety and tolerability studies. It's an allosteric activator. Allosteric activators are not easy to make. It's easier to say it than to make it because you're not optimizing for binding the enzyme as you would with an inhibitor typically. You're optimizing for a molecule that binds and changes the conformation of the enzyme in such a way as to make it more active. So we've spent a lot of time on medicinal chemistry, optimizing the activity of the compound, the assays, and also optimizing the physical properties of the compound. We have many studies done. We like to do all our cellular studies in human cells, so we primarily work with human iPS neurons and patient-derived PBMCs. And we like to use mice for PK/PD studies only, not to try to de-risk efficacy, but just to show us that we can get a dose and exposure in the brain and that it can activate the enzyme in the brain. And so we have all that data, of course, in advance of the beginning of the first-in-human study.
Thank you. And Fabrizio Stocchi?
Good afternoon, everybody. I'm professor in neurology at the University San Raffaele of Rome, and I've been involved in Parkinson's disease since 1983 in different clinical trials. I'm a neuropharmacologist by PhD, so we conducted a number of trials and try to find a way to make a disease modification. Actually, we get all these ideas here, and then we put them on the ground, actually, and we try on patients, phase 2A down to phase 3, and most of the time we fail. And so far we always failed in demonstrating a drug that can actually really change the course or the natural course of the disease, even if there are some signals that actually using drugs in a certain way, we can actually change the course of the disease, but nothing really what we call neuroprotective, even if today we prefer to say disease-modifying action. Anyway, the last study we were involved was, in fact, for neuroprotection with the new LP1 drugs, and again, it failed. And then years ago, we had the nice surprise to see a drug, which is rasagiline, showing a disease-modifying effect in a clinical trials. But from that trial, we learned a lot, even if we don't know if that was a real disease-modifying effect. We learned how to design a trial, how can we adjust the clinical trial. We found that there are a number of problems in demonstrating disease-modifying in Parkinson's disease, starting from the scale to the lack of biomarkers that actually are reliable in PD follow-up. So this is the clinical side of the story, which is probably less exciting than basic science, but this is where we should actually demonstrate that the drug is working on patient.
Walter Koroshitz?
Hi, I'm Walter Koroshetz. I'm the director of the National Institute of Neurological Disorders and Stroke at NIH, and I work with Eliezer pretty closely on the neurodegenerative diseases. And so our institute is a major funder for research in Parkinson's disease. About, say, 20-25% of our budget goes to real basic neuroscience, that's disease agnostic. About 40% goes to trying to fund research on mechanisms of disease, so mechanisms of Parkinson's disease. And then we have a virtual pharmaceutical-like company to move therapies from the bench to the point of an IND, or even better, to a company taking the drug off our hands. And then we also have clinical activities, so we're currently funding 2 trials, phase 3 trials in Parkinson's disease. One is based on some epidemiologic evidence that people taking calcium channel blockers that get into the brain had a slightly decreased incidence of Parkinson's, so we're testing isradipine. And the other one, similarly, is inosine to elevate urate levels because of, again, epidemiological evidence that people with high urates have less incidence of Parkinson's disease. From the trials that we're doing and the trials that we have done in the past, none of which have worked, we're certainly been very sensitized to the dangers of jumping from our very minimal knowledge to actually get things going to work in people. And so we're always trying to kind of fill in those gaps of knowledge to make the jump to a clinical trial a higher probability of success. A number of years ago, it came to the forefront, and I'm sure it's in everyone else's mind, that one of the big drawbacks is the lack of biomarkers that can be used for developing signals for target engagement in Parkinson's disease. And so we did initiate a fairly significant effort to develop biomarkers in Parkinson's. We have a sample collection of CSF and blood and urine. We have numerous discovery grants out trying to develop biomarkers. And we're hopeful that, combining the advances in basic science, a really good biomarker that we can follow as a therapeutic target along, that will have a higher probability in the future, either with NIH funds, but actually we'd prefer if the industry will use the funds for the phase 3 trials, as long as they work.
Actually, Serena, maybe you could grab that mic and we can keep that one down there. This is Serena Hung.
Okay, hello. So my name is Serena Hung. I'm not Jessie Cedarbaum, and in case you're confused. I work at Biogen. I'm the medical lead for the Parkinson program. By training, I'm a clinician, a movement disorder specialist. So I echo a lot of what Fabrizio and Walter was saying, that we have learned a lot in clinical trials, but we still got a lot to learn. So in my day-to-day job, I lead the clinical trial portion of the program for an anti-alpha-synuclein antibody. And currently, the trial is in phase 1, and we'll be presenting some healthy volunteer data on Sunday. But, my other panelists have said, even though we've learned a lot, and Jamie has said, we thought it was easy, but it's really not. Part of my job is to figure out what tools I have in order to help us demonstrate the disease-modifying effects of a drug. And also, one of the things that I think would be important is if something doesn't work, can we figure out why it doesn't work rather than going blindly into another clinical trial? Patients' lives are, very precious. Their time is very precious. When they volunteer for clinical trials, they're actually giving up a lot. So we need to really get good at our jobs in order to not waste their time and get a therapy to them as quickly as possible. So at Biogen, we actually believe in advancing biomarkers. So we actually have some pretty extensive efforts in looking at various modalities. And we work with a lot of different groups, including the Michael J. Fox Foundation and other consortium, in order to advance this effort. So hopefully, at some point soon, we'll be able to develop a drug that can demonstrate disease-modifying therapy. But once again, we need the tools. We need to be able to figure out, that it is a disease-modifying therapy when we have one. So, there's a lot of exciting work that needs to be done.
Jim Galvin?
Hi, I'm Jim Galvin. I'm a neurologist. I'm a professor and associate dean for research at the Schmidt College of Medicine at Florida Atlantic University. This is one of the newer medical schools. I was recruited to come down here to set up a neurodegenerative disease research center and clinical trials unit. My research is really focused on the non-motor and cognitive features of neurodegenerative diseases. It's focused largely on detailed patient characterization, clinical pathological correlation, clinical trial development, and developing diagnostic tools that could be used in the community. However, the role I'm here today is I also serve as the chairperson for the NIA work group to develop a Lewy body dementia module to be used across the NIA-funded ADC programs, as well as NINDS-funded programs that are characterizing Lewy body dementias. And so this work group came up with a module that'll hopefully go into effect right after the directors meeting in April. And it really is going to characterize the motor and non-motor features associated with Parkinsonism, the sleep, attention, fluctuations, neuropsychology, genetics, and biomarkers associated with Lewy body diseases. And this will allow us to harmonize efforts across the Alzheimer and Parkinson world to better understand the changes that occur in Parkinsonian disorders and really develop a trial-ready cohort that's going to be available to do these exciting clinical trials we're going to be talking about.
And Gene Kinney.
Hi, everyone. I'm Gene Kinney. I'm the CEO of Prothena Biosciences. Prothena is a company that spun out from Elan Pharmaceuticals back in 2012 with many of the drug discovery assets from that company. So one of our main focuses is around disease areas that are thought to be caused or where the progression is involved with misfolded proteins. And so we focus both on peripheral amyloid diseases as well as central amyloid diseases. We do, like my colleague here from Biogen, have an anti-alpha-synuclein antibody that is in clinical trials today. We'll be reporting on Sunday. In fact, Dr. Joe Jankovic in the audience here will be talking on Sunday about the phase 1b multiple dose study where we've evaluated this molecule, PRX002, in patients with Parkinson's disease. So we look forward to sharing those data with everyone this coming Sunday. I think the only thing I would add is just I'll make an unequivocal statement that I agree with my panelists here that disease modification and curing that in Parkinson's disease will be hard. I'll definitely concede that point right up front. But I think I'm looking forward to a very productive dialogue here today and looking to learn from my panelists.
Wonderful. And I just wanted to mention that Jose Obeso was supposed to be on the panel today. He was unable to come to the meeting because of some travel issues. So we will miss him, but we'll be thinking of him. I also was so excited to introduce the panelists that I realized I didn't introduce myself. I'm Jamie Eberling. I'm one of the scientists on staff at the Michael J. Fox Foundation. So now I want to jump right in with your questions. And if you want to direct your question to one of the panelists, please do, or you can just direct it to everybody and let me pick. And to the panelists, if you're dying to answer a question and I don't call on you, feel free to wave at me and I will call on you. So let's get started with the first question. Would the multimodal approach, including non-pharmacological interventions that seem promising for Alzheimer's disease, also be a preferred option for Parkinson's disease? So maybe we'll start with Jean.
Yeah, I mean, I think ultimately these are complex disorders, I think, to state the obvious. And I think, thinking of holistic approaches that treat the disease in a multimodal fashion makes perfect sense. Obviously, as a biopharmaceutical company, we believe that the underlying biology is something that has to be dealt with concurrent with other approaches. But I think that there's nothing to say that a multimodal approach wouldn't be preferred. Maybe I can defer to some of my more clinically oriented colleagues here.
So I think there's an expression, all roads lead to Rome. And so if you look at a map of the Roman Empire, you can much start from any large city and follow a Roman road, and it'll lead you to Rome. Likewise, there are probably multiple ways to get to many common neurodegenerative diseases. They share a number of traits and features. And when we're talking about these concepts, it's not just genetics and pharmacogenetics and drugs, it's lifestyle, it's family history, it's comorbid medical conditions. So really a multimodal approach is probably the only way we're going to get to really make a significant impact on the patients suffering from all of these neurodegenerative diseases.
So I'm in agreement that I think a multimodal approach to treat a patient is very important. However, it actually brings up some issues with regard to clinical trials. Because, when we want to demonstrate the effect of a drug, we actually want to control as many variables as possible. And a lot of you probably know in human trials it's actually really difficult because there are a lot of things that we don't know about in a patient's daily life. And so how do we control for that? So I think, while actually later on when we actually have these therapies and we put them together, we actually need to properly study it and say, what works, what doesn't work. But when we're actually doing a clinical trial, it actually adds a lot of complexity. So for example, one thing that comes to mind is, there's a lot of reports about exercise being beneficial to Parkinson's disease. And yet when we're in a clinical trial, we don't necessarily control for that. We're not very good at controlling for that. So when we see some, kind of uninterpretable result, how are we going to deal with that? So this is actually something that I think the community will have to learn how to do when we're planning the clinical trials.
Right, so I guess there's a lot we don't know about how the environment interacts with the brain in terms of Parkinson's disease. It's one of the most interesting areas of research now, I think. But I would go back, the thing that's been studied most has been exercise in terms of really interesting findings. The University of Pittsburgh folks actually showed exercise reduced MPT lesions in monkeys. We did a study, funded a study showing that Tai Chi exercise improved functional outcome in people with Parkinson's disease. And I think also, I guess, one of the big areas now that's opened up is the interaction between the microbiome, the enteric and autonomic nervous system in patients with Parkinson's disease, because they're potentially a two-way process there whereby toxins may be coming in through that system, the system's affected, and so the digestive system is also dysfunctional. So there's lots of different factors. Some of them are related to symptomatic benefits. I remember when I took care of patients, I had a rabbi who would exercise and not eat before on Fridays, and that would make him very highly functional for Saturday, and then he would eat, and then he would have really bad Parkinson's the rest of the week. So there are ways, lots of things that can affect the symptoms.
Well, first of all, I would like to say what I believe that exercise is the only thing which has been demonstrated in Parkinson's disease to improve patient. But I wanted to be very clear. The exercise without drug intervention will never cure PD. And any other approach, which is non-pharmacological, can help, but definitely cannot cure PD patients. So, and I'm saying that because the web is full of crazy things, diet, aliments, vitamins. Nothing has been shown to be helped to help patients with PD seriously. So the drug intervention remain the most important side of the story. And then we can discuss what we mean for drug intervention, which kind of drugs we consider, or if we believe that everything is drug intervention. That's another story. The other thing which I want to ask everybody is, what is Rome? I know what is Rome because I live there, and all the roads come to Rome, that's for sure. But in Parkinson's disease, what is Rome? Everything leads to Rome, but this was something I wanted to ask you. You work in different pathways, but what is the end of this story? Where all this pathway go to? Is it alpha-synuclein? And then we are on the right target. But if it's not, what is actually Rome? What is Rome?
I don't know what's wrong, first.
Well, you should come and
but I would say, I would bet it's not always synuclein. Strongly bet that. And that's because, the risk associated with synuclein, while very significant, is really small, right? It's just one ingredient that contributes to sort of a mixture of problems that if you have 3 or 4 of these problems, then you'll develop disease. If you have 1, maybe not. So I think the important thing is that we have to recognize, and this is coming from not a neurologist, not a physician, that Parkinson's disease is not a single entity. And even at the clinical level, my listening to clinicians has convinced me that there are multiple entities. There's really no such thing as an average patient. At the pathological level, there's no such thing as an average patient. And I would like to see a lot more effort spent on trying to understand, which group of Parkinson's disease suffer from abnormality X, then those could be maybe treated, and then which group suffers from abnormality Y. In our case at LTI, we've been given this gift of this mutation, which is easy to genotype in patients, where we can pick out those patients who clearly have that abnormality. But I think in other situations, I think that there has to be a lot of work spent on defining subtypes of disease and just throwing away this 200-year-old idea that it's all one entity and that everything has to converge, because I don't believe there's any evidence that it will or that it does.
And Eliezer, since everybody else answered that question, you're going to have a chance to. It was a multimodal question, and it required a multimodal answer. So, Eliezer, do you want to comment on that?
I mean, I would say that bottom line, I think since we're talking about disease-modifying therapies, I mean, the bottom line is that we have to come up really at first, and I completely agree with you, that we need to come up with a specific target and a disease-modifying approach at a very early stage, and whatever else we do afterwards, sure, it would help, it would be useful, but bottom line, we really need to find a drug that would specifically target a pathway at very early stages. And again, since we're talking about disease-modifying therapies, we're talking about primary prevention, secondary prevention type of trials. We're not really talking about patients with mild to moderate disease where a considerable amount of the dopaminergic system and other systems are already wiped out. I mean, we're really talking about preventive trials. And these are trials that are not a one-year trial and a two-year trial. As we have learned from the Alzheimer's disease field, these are trials that are going to take 4 or 5 years, maybe even more than that. So we're talking about also funding. And how are we going to fund all those long-term trials to really test the question of disease modification? I mean, and I agree with Peter, the target could be synuclein or could be other things. I'm totally open to whatever is your target for disease modification, but these are not going to be short-term trials. 1 year is not going to make it. So I think we really need to think about, because often we make the mistake of saying, well, the clinical trial failed and the drug didn't work, or the amyloid hypothesis is wrong, or the synuclein hypothesis is wrong. Well, it's not that. It's that we're testing a disease that is a long-term, very slowly progressive disease in a very short window of opportunity without, as Walter was saying, without appropriate biomarkers. So I think here the problem is we should not repeat the same, let me put it in a different way, in a more positive way. We should learn from what clinical trials in other neurodegenerative diseases have taught us. I'm really excited about the synuclein immunotherapy approach, which is something that we work together with Gene and his group and others. But I know that if we just throw that antibody into patients that are in later stages of the disease and we don't have a long-term primary or secondary preventive study, I don't know, we might repeat the same story as what we have seen in Alzheimer's disease. So I think that's something we really need to think about. And then, sure, if other modification, exercise, this and that, and that will come later, sure, that's great. But I think we have to address at first that very primary point.
Let me ask you all.
Fabrizio, and then Jean has a comment as well.
A couple of questions about what you said. First of all, I think that Parkinson's disease is different from Alzheimer's for many reasons. And one of the reasons is that we have more time. I mean, first of all, we have at least 10 years before the motor symptoms appear. We have to discover the patient, that's another story. But let's say like that. We have 10 years before. Then the motor symptoms appear. Then we have another 7 years for the dopaminergic neurons to disappear. And then we have probably another 15 years when the patient fight with the disease in different terms. So my points are two. Is it possible, well, first of all, should we focus on alpha-synucleinopathy, right? So is it possible that, for example, we should change intervention according to the timing? So let's say that the disease starts with a different mechanism, as you proposed, and then arrive to alpha-synuclein. Now the alpha-synuclein become the target. And now I go to a more, let's say, symptomatic effect on alpha-synuclein. And there are two different interventions, and it might be that alpha-synuclein is important also later on. So it is possible that we can intervene in a different way according to the timing and according, of course, to the patient's characteristics.
No, I don't disagree. I mean, I think on that topic as well, it's important when you think about timing that we talk about the disease itself and what it is. You talk about the motor symptoms, but of course we now, I think, understand much better that there are premotor symptoms that may be components of the disease itself and not just prodromal symptomatology leading to the disease itself. And to the extent that synuclein may be involved in some of those processes, whether it be, the fact that, 80% plus patients have constipation, which many times is seen years before the motor symptoms come about, the well-described REM sleep abnormalities, hyposmia. I think, what we're learning is that it's a complex disease that has both motor and non-motor components. Some of those premotor components are part and parcel of the disease itself. And so how we think about treating and delaying disease and when we intervene may certainly be influenced the more we learn about these components of the disease.
Okay, Peter Lansbury.
Oh, you're going to ask me a question?
I was going to ask you a question.
I just want to argue with Eliza here. I do agree, right, that run-of-the-mill average Parkinson's disease is very slow. That's the problem that we fight with Alzheimer's disease. It's the opportunity in that if we could just make it a little less fast, we'd make a big difference, but it's the problem and then the trials are long. However, it's not, if you segment the disease, it's not true that all populations progress at identical rates. So if you look in the patients who carry this GBA mutation or polymorphisms, of which 10% of in all US studies now, 10% of patients have this, their progression rates are significantly faster than typical Parkinson's patients. And depending on the genotype, those progression rates can be extremely rapid. So we're guessing at LTI that in that population, we can actually observe a disease-modifying effect in a 1-year dosing trial. Now, the timing of the initiation of dose is another issue that's been well discussed here, but as with about 100 patients per dosing group, as opposed to the typical trial, which would be 18 months to 2 years and would have hundreds of patients per group. That is a huge savings. And if we keep suggesting 4-year trials with 5,000 patients, patients will run out, both kind of patients, right? It puts a huge strain on the patient population, but also the patients that pharma so far has shown in the Alzheimer's field, I think will run out if we don't make an effort to find groups where the progression rate is significantly faster and we can start to make a dent one little piece at a time in the disease.
Okay, I was going to ask you a question, but I'm going to hold it for now, and I'm going to ask Jim a question, and then probably I'll have Eliezer comment on this also. Which animal models of Parkinson's disease best correlate with one or more of the human phenotypes, and do any of these model the cognitive defects in PD patients? I think I know what you're going to say.
That's a great question. That's always what we start off when I don't really have a good answer for you. So, the challenge with all animal models is they generally only recapitulate small aspects of the disease. And so the simple fact is that rats aren't people, although some people are rats. So, you can only take a certain amount of the information gained from any one animal model and then figure out what that relates to a clinical trial. That's why we're very effective at curing mice and rats and flies and worms, and we're not so great at curing people because the animal models simply just don't recapitulate the entire aspect of the disease. If you think about Parkinson's disease as solely a movement disorder, then you're really missing the full spectrum of Parkinson's disease, and the animal models just can't copy that. And so that's the great challenge, whether you're talking about the motor side or the cognitive side or the REM side or the enteric nervous system side. Parkinson's is really that entire picture. And so you're taking a incredibly heterogeneous disorder and trying to cram it into a homogeneous definition in order to run a clinical trial. And that's where I think some of the great challenges are.
Eliezer, do you want to comment on that as well?
I come from a perspective of animal model development, and I was here 30 years ago when people say, well, it's impossible to develop a genetically modified animal model of a neurodegenerative disorder, where, when the first mutations and synuclein and APP and tau and other molecules involved in neurodegenerative disorders were discovered, and the first conventional, let's say, transgenic animal models were proposed. I mean, a lot of people were very negative about them, and they said it was impossible to develop these type of models, and obviously that was not true. A lot of these models were developed, but I think there was always the idea that overexpressing some of these either wild type or mutant form of these proteins were going to recapitulate the whole spectrum of disease, and as we have learned over the years, that's not completely true. But it's a classical half full, half empty glass of water story. It all depends which perspective you look at these animal models. And I think we have learned tremendously in terms of understanding the pathogenesis of the disease and the disease-modifying genetic factors of the disease from the animal models. Sure, we have learned less in terms of how to develop drugs based on the animal models. And I agree with Peter that probably utilizing cell-based human models and so on might be equally useful or might be complementary. But I think what I want to stress is that coming from the perspective that I come, I think that a lot of what we have learned in terms of the natural history and the pathogenesis of the disease, Parkinson's, Alzheimer's, and others, have come from modeling in animals, and this has been tremendously valuable. Now, how do we use that in terms of developing drugs? I think it's a mystery at the moment, no? But I think there is a lot of smart young people here in the audience that we'll figure that out in the next few years. I think with the new technology, with the CRISPR, and with the new approaches, I think we'll get better models that will give us better answers.
So, we do actually have great animal models that we don't really think about as being animal models, and that's human populations that have mutations or polymorphisms that massively increase their risk or causative of the disease. So a lot like Peter said, instead of doing a trial in a general Parkinson population for 10 years, you might be able to select a group of people who have a specific mutation that make them rapidly progress. I mean, this is what we're doing in Alzheimer's disease with the Colombian PS1 mutation, with the Diane-2 studies looking at people with PS1 and APP mutations. And so it's possible that we might get our best answer not from thinking about what we learn from animal models, but we really think about looking at selective groups of genetic forms of disease and trying those medications in there. Since they're going to get disease at a certain age, depending on the family's makeup, then if you started a drug 2 years before that and they reach their expected age of onset and don't actually develop disease, then you've modified the disease. And I think we can answer the question not in a simple way, but in a logical way by thinking about using more human models of disease.
Well, I partially agree what you said. I mean,
I only partially agree too, so.
First of all, in Parkinson's disease, we can easily demonstrate a disease-modifying effect in a year. I mean, early patient with Parkinson's disease not treated progress 10 points in the UPDRS. Now we have thousands of data showing that. And if you have disease-modifying drug, you can show a slowdown, a progression of the disease and on the UPDRS. And UPDRS is not the best tool we have to score patients. So that's the limitation because of the floor effect and so on. But still, you can do it. So that's the first point. The other thing is about the genetic form. I agree entirely that the genetic form, they progress more rapidly. LRRK2 is even worse than GDA and so on. But are they the patient with Parkinson's disease? I mean, so far, 22% of the patient, the entire population of patients have a genetic form. What about the rest 80%? So we can focus on a genetic form, but we address that kind of treatment on that genetic form. Whereas if you try to look at the entire population of patients with Parkinson's disease, excluding probably the genetic form, you actually address your treatment to a much larger population. But I know that is much more difficult. But today we have approaches. We have adaptive trial. We have disease-modifying delayed start design trials, which actually can give us an answer in a relatively short time. And if the drug hasn't got any symptomatic effect, you can see something in 6 months.
Okay, I guess Walter and then Serena.
Yeah, so maybe I was going to say what you're going to say. But so one part about the animal models I'd emphasize is the fact that what you really would like to get to in the animal models is understanding the pathophysiology and how to target that pathophysiology. And that's what there's been a lot of effort on. But I think to pay off in the human, the next step is to try to develop measures that you can get from your animal model and potentially related to what your drug does, that then you can measure in the human. And so in my position, we see all bunch of different diseases, and I can tell you the ones that progress the fastest in terms of therapies are ones in which there is a human biomarker that you can target. So multiple sclerosis, which is going to go off to Biogen, my understanding is difumarate was, not really even went into animal models. It had a known pathophysiology mechanism, and the biomarker of enhancing lesions in multiple sclerosis is such a well-worn path towards approval of a drug that all you needed to do is to go into patients, see if you could prevent those lesions from developing, and you know pretty well you're going to get a drug to market. So that's kind of at the end of the rainbow, I think, for Parkinson's as well, is to get something that you can really hang your hat on, that you're affecting the progression of the disease. You never know until you get a successful trial, but once you have something that targets the biomarker and it's successful, then it's just going to be one better drug after another. And so I would encourage people in the animal model world not just to go after pathophysiology, but think about what can you measure related to what your drug is doing that you can then apply to the human case.
So Walter did say some of the stuff that I was going to say. But actually, one thing that I wanted to expound on is actually one thing we struggle with a little bit is, the predictive value of these animal models. Right now, we don't have a success in disease-modifying therapy. Once we have a success, we can actually go back to the animal models and figure out, what are these animal models really telling us? And until we get there, right now, there's a lot of guesswork. Every piece of animal model data is basically giving us another piece of the puzzle, but ultimately, how does it translate into the human? We don't know. So that's actually is one of the problems. Then the other point that I was going to make was actually responding to Fabrizio about, demonstrating an effect easily in a short time. I think one of the things that we learned from the previous trials is the complexity about symptomatic treatment and how it actually make the picture very cloudy in demonstrating an effect. So, theoretically, if we can actually keep patients off of these symptomatic treatment for long enough, then maybe we actually can get a clearer picture. But in this day and age, when the symptomatic treatment, especially in early Parkinson's disease, works so well, I don't think it's actually that feasible for us to keep patients off medications. So in order to demonstrate disease-modifying therapy effect, I think we need to really figure out some biomarker maybe that actually is perhaps less affected by symptomatic treatment, or maybe actually the biomarker actually is in a completely different pathway as the symptomatic treatment, so that it's the two of them will not, you won't mix the effects together. But those are some of the very practical issues that when we're doing clinical trials, we really have to plan for and understand.
Okay, Peter?
Yeah, I just want to come back to one point that was raised earlier about the 80%. Of course that's true. We want to treat everybody. But you have to have a success first. And I think if you focus on the 80%, which is sort of what the Alzheimer's field started doing, they're now 15 years in, and they're down to ApoE4 homozygotes, which is maybe 2%. So, and that's where they're going to get their success if they get a success. The other point I would make, though, is, and again, of course, it relates to, I want to shill for the company. But with the GBA mutation that decreases activity in the GCase gene, we understand now that in the wild-type population, patients who do not have a mutation, activities vary widely. And I would say from what we've seen and the reason we know so much about this is because this is an enzyme assay that's done in newborns to test for Gaucher disease. And so we know that probably 10% of the wild-type population, the normal population, has activity less than 50% of the mean. So that's an incredible variation in the human population. What we think at LTI is that there are going to be many patients who don't have a mutation in GBA but have a biochemical abnormality whose consequence is the same as a mutation in GBA. We call that a sphingottype, and we've been going out and trying to find those patients. So you start with the genetically defined population. You understand what it is that makes that mutation bad, because of course it's not the change from a G to a C itself, it's something downstream of that. And you find out why that's important, and then you go and ask, in the wild-type population, can we find other patients who don't have the gene but for complex genetic reasons may have that same abnormality? So I think that, again, I'm still voting for trying to find a niche where you can run that first trial, have a success, and then start to chip away at the disease, because there probably are a lot of patients out there who would benefit from GCase activation in addition just to the mutant patients.
Okay, Jean, I have a question for you. Given that Lewy bodies are intracellular, does an immunotherapy approach even make sense?
Yes.
Okay,
More?
it's got to be longer
Okay,
than that.
All right. So, no, it's a great question, and I will tell you, working with Dr. Maslia, the original experiments here started all the way back in 2003, and I think a lot of the work that was done moving from those original experiments to the first in human start with an anti-alpha-synuclein antibody, or at least our anti-alpha-synuclein antibody in 2014, was much revolving around that question. How is it that we're seeing the effects that we're seeing, particularly in the various transgenic models where these antibodies had been tested, reduction of pathology, which in large part is intracellular, but also then downstream effects in terms of protection of synaptic integrity, behavioral effects, and what have you. And we spent a lot of time thinking about that question. I think one of the aha moments for us occurred probably in the late 2000s when the idea of the cell-to-cell transfer of these misfolded proteins started to take a little bit more hold. And at that time, we had a number of antibodies that targeted different epitopes on the alpha-synuclein protein, targeted different post-translationally modified forms of alpha-synuclein. And what we started to learn was that there was a relatively good correlation for us between those that did a good job at preventing that cell-to-cell transfer and those that were showing good activity in the animal model systems in an in vivo setting. And so it led us to believe that minimally there's a epitope that is conserved on the forms that are capable of transferring from cell to cell and those that are more involved, at least in the pathology across the multiple animal models where we had investigated, notably the line D and the line 61 mouse. So we thought that was quite interesting. I think, we have done a little bit of work, and in fact, Dr. Maslia, you may want to speak to this. He did this work looking at fluorescent labeling of some of these antibodies. You can see this co-localized with cathepsin D inside the cell, so it appears that these antibodies do move into the cell. What exactly they're doing in the cell, I can't say with any certainty, so it's a little bit hard to understand exactly what that mechanism is. I think so where we've kind of settled on this is that we think one important mechanism from an immunotherapy perspective, particularly those targeting alpha-synuclein, is around the inhibition or prevention of the cell-to-cell transfer process, which we think is, the science is quite good, supporting that plays an important role with respect to progression of the disease, particularly involvement of different brain areas and the symptoms that are involved and come along with that. But we're seeing clear evidence that we can remove intracellular pathology as well. And although we don't fully understand those mechanisms, we can't discount the possibility that there may be some intracellular activity of the antibody. We do see it in there. Again, I just can't tell you with certainty what exactly it's doing in there.
And Serena, do you want to comment on that also?
Sure. So actually, I'm largely in agreement with you as well, that there is a place for
That's no
immunotherapy.
fun. We want to have some controversy.
I know. No, so, because actually, I joined the program, when a lot of these animal experiments were done already, and I focus on the clinical side. But, looking back at the experiments that have been done, and actually my colleague, Andreas Weihoven, has actually presented some of this data here, that we're actually seeing some of the same type of evidence to support that by targeting the cell-to-cell transmission of alpha-synuclein, it does have an effect. So I think at least I have quite a bit of confidence that, we're affecting something that is important in the disease process. So, we're moving forward based on that confidence.
Does anybody disagree? Everybody agrees?
Yeah, but I
thought this would be more
I have
controversial.
A curiosity just coming out in my mind now, and I ask them that have done all this work. Did you look if using the antibody, the antibody attack, of course it should be, but attack the alpha-synuclein also in other parts of the body? And is it easy, for example, to find out if disappear from the intestine or the salivary glands or so on?
Yeah, so you're asking if, for example, the antibody would interact with synuclein in an enteric nervous system, for example.
Intestine,
Yeah,
for
I mean,
example.
we expect that that's absolutely the case, particularly with peripheral administration, where you're going to have your highest concentrations in the systemic circulation. So that makes perfect sense. I think, I don't know if in the animal models we've specifically investigated that, but certainly it's logical and makes perfect sense.
Because that would be easy. That would be easy to find out if the drug is really working. Just, for example, biopsy the intestine, the colon, which is very easy.
Yeah, we've looked at that. I know colon biopsies are something that's been thought about, and there's been a lot of work done with respect to doing that as a biomarker, and potentially for different reasons. One would be for inclusion-exclusion, to make sure that patients actually have some level of synuclein. I think we worry a little bit about the biopsy-to-biopsy variability with respect to absolute quantitation. So in order to use that, I think from an interventional setting and actually ask the question of whether you're seeing less synuclein over time, you need to make sure that the sample-to-sample variability is quite tight. And at least from our perspective, we haven't been convinced yet that it's tight enough to be able to do those kinds of interventional studies. But certainly, I think from a inclusion-exclusion, enriching your population perspective, those are important biomarkers to take a look at.
I guess the question with the antibodies is always access to the central nervous system and effect size. So when you're trying to sop up something, you want to have a pretty, almost a large concentration. And so you're still working in the less than 1% range?
So we'll be talking about those data on Sunday, so I don't want to talk about the data before we're ready to talk about it. But it is, at least in our multiple dose study where we did CSF evaluations, we saw levels that were at the higher end of our expected range, which was positive. I'll also say that in the transgenic mouse studies that were performed, those were systemic administration, intact blood-brain barrier systems. And at least with respect to clearance of pathology, our minimum effective dose was typically less than 1 milligram per kilogram.
But people are working on ways of getting antibodies
absolutely,
in better.
and I think
that could potentially be a game changer.
I think those are smart approaches, and I look forward to seeing those work. I will just say one thing, and I think my colleague from Biogen will probably agree with me again, but which is that we think one of the important characteristics of these antibodies is that they do have a high preference to more aggregated forms of the alpha-synuclein protein. We believe that that's the more pathological form. And so by building that into your antibody, in the case of our antibody, for example, we have over a 400-fold avidity shift to the more aggregated forms. And by doing that, what we believe it allows us to do is minimize the dwell time in compartments like the periphery and potentially in the CSF, where we expect there to be less alpha-synuclein, and increase the dwell time in the central nervous system, where we think the activity really needs to be.
Yep, so Can I respond to that just because, there's actually some similarities in our thinking. So we're also presenting our data on Sunday, so I'm not going to speak about that, but I can tell you that the CSF penetration in our human study is also as expected. So so stay tuned on Sunday. So the other point that Jean made was about the aggregated forms of alpha-synuclein. We also believe that we want to target the aggregated forms, and there's the belief that there's not as much aggregated forms of alpha-synuclein in the periphery, so hopefully it will bypass, or bypass the periphery and drive the binding more to the CSF or the central nervous system where the appropriate form of alpha-synuclein is present.
Eliezer?
Yeah, just to be a little bit provocative, you asked if there was any dissent, and of course, it's hard for me not to agree that immunotherapy works, and I think most people here have proven maybe the mechanisms, etc. There might be, but I think the question that we need to ask, and again to be a little bit provocative, is either if we select cellular, because that's the other approach, cellular immunotherapy, or if we select antibody-based therapy, either vaccination or passive immunization or what have you. What are the, and we know from the phase 1 trials that at least in that interim it's safe. What are the challenges of these different modalities of immunotherapy when using them over a long period of time? Because again, I'm going to come back and I'm going to disagree. I think you have to think long term for these neurodegenerative diseases, and you need to conduct long-term trials. So what are the challenges with immunotherapy? I mean, for me, with at least with alpha-synuclein as a target, there is a really interesting problem, which is that we have learned in the last few years that alpha-synuclein actually plays a role in regulating T-cell maturation, and probably even B-cell maturation, and it plays a role in the immune system. So, and actually we know that, for example, from alpha-synuclein knockout mice and other models, that they have T and B cell maturation problems, etc. So when we're utilizing, for example, active immunotherapy approaches or T-cell-based immunotherapy approaches, and we have a synucleinopathy, how is that going to interfere with immunotherapy? Or, for example, when we use these therapies for a long period of time, do we develop autoantibodies that are neutralizing to our antibody of interest? Or what are the challenges of these type of therapies when we do long-term treatments? And I think that that's where the interesting question is. But I think, definitely that it works, but what are we going to do in the long term? I think it'd be good to start thinking about that.
So I just want to be provocative in a different way. So it's not against the thought of using immunotherapy, but I think one of the unanswered questions that we have is, if we use an immunotherapy and remove what we think is the problem, does that necessarily mean that the patient gets better? So I think what we've learned, at least a little bit in the Alzheimer's world, is that you can remove amyloid, but if you don't do it soon enough, it may not work at all. So once people actually have disease, removing the offending protein may not solve the problem. And so this could very well be true for Parkinson's disease as well. So not only do we have to test the question, but I think we have to figure out a better way of figuring out who is at risk for developing Parkinson's disease. And this gets to the question of having a biomarker that really tells us something about the fundamental aspects of the disease. So once you have motor symptoms, it's possible that removing alpha-synuclein isn't going to fix the problem, just like removing amyloid doesn't fix the cognitive disorder once you manifest the symptoms. So I'm all for immunotherapies, but I think you can have the right drug and the right target, but at the wrong time, in the wrong population, you're still going to get a negative answer.
Okay, that was a good segue. I want to ask a question about biomarkers. First of all, what do you think are the most promising biomarkers that we have today that can be used in disease-modifying trials? And one thing I spend a lot of time worrying about is that we have these exciting treatments going to the clinic or getting close to the clinic, and they may be doomed to fail because we don't have the best tools to use in the clinic. So I'd like to get maybe all of you will have a comment on this one. Walter, maybe we'll start with you.
Well, I think that's our big problem right now, is that we don't have anything we can hang our hat on. I think we look at, what's going on in Alzheimer's disease with the tau PET marker is, again, it's intracellular, so you'd like to say, okay, well, that's what we need, synuclein PET marker, but that's been really difficult to do, as you know. And
I know better than anyone.
But, it's something that would change, that would be a game changer if we get that going. I was pretty impressed today at the biomarker session about the CSF studies, being able to pick up seeding of CSF and synuclein in people with Parkinson's disease. So it's early on, but something like that where you can follow patients over time with CSF examinations, potentially, again, for a synuclein target might be something quite good. But I think that's where we got to put our big push now.
Peter, do you want to comment, and then Fabrizio?
Okay. Well, I mean, there's two kinds of markers. I think one, the first kind, and the one we're obsessed with and have been obsessed with, is the proof of mechanism marker, to show your drug's doing what you say it should be doing in the brain. And that's absolutely a requirement. So that depends on the drug. So obviously we'd have a different proof of mechanism marker than Gene would. But the next marker, I think, should be a marker that increases your faith that efficacy will result from your drug doing what you say it's doing in the brain. That's really hard. And, from our perspective, we've been looking at things that are out there because, developing a de novo marker doesn't make sense for a company like us. It probably doesn't make sense for anybody to be ready in 2 years. So we've been looking at, things like FDG PET, EEG, things like that, where if you see a signal early in the trial, will that increase your confidence that you should keep running on that trial? I mean, my particular bias right now is no. Because I think there's nothing that if you didn't observe it, would you kill the trial? I mean, you have to ask that question. Like, if I don't see that FDG PET signal, I'm stopping the trial. And there's nothing out there like that. So for now, we're just waiting for clinical signs. But I do agree that would be a huge game changer if there was something that you could really put your faith in. Ideally, that was like with the synuclein case, it's an ideal situation where it's related to the mechanism of the drug, where you could make that decision. You would have a decision point, and if you didn't see a change, you would stop the trial.
Fabrizio.
From the clinical perspective, well, I agree entirely with Peter. I mean, if you're looking at, first of all, I think we have to distinguish what we mean for biomarker. I mean, do you want a biomarker to follow up your trial and your drug, or we want a biomarker to make an early diagnosis and follow these patients? These are two completely different aspects. And a biomarker like sugar in the blood or blood pressure or ECG has to be very easy. I mean, we have to find a way to spot patient, let's say, a risk of. No, I don't even say with prodromal Parkinson's disease, but a risk of. We tested a number of things, nothing really worked, but eventually, probably putting together all the non-motor symptoms, we can actually build up an algorithm and see if this patient, then we know that the SPET scan, for example, can spot a patient 5 or 6 years before the motor symptoms appear. So that could be a biomarker, clinical biomarker. Then we go into clinical trial. And then the story changes because you can actually ask the patient for something more difficult, like a CSF, which I don't see a clinical general biomarker. But it can be a biomarker in a clinical trial and the same with the PET scan. And at the moment, I think what is closer is probably the neuroimaging are closer to be a biomarker in clinical trials. We'll see about CSF because the results of the CSF and alpha-synuclein, I saw the results of the big American trial on patients, 1,700 patients presented it 2 days ago, and they found a low level of alpha-synuclein in the CSF. What does it mean? So, I mean, I think we are far, but the good news is that in Parkinson's disease, clinical sign, if they are well scored, better scale than UPDRS, which probably take into account also non-motor symptoms and so on, it's actually reliable. It's linear in the first 5 years. It actually, it goes together with the change in SPECT scan. So still we can score and follow up the patient from the clinical point of view quite in a reliable way. But we definitely need a biomarker. We definitely need a biomarker, that's for sure. And probably in your population, the GBA will be easier because you know what you're going to target. The problem is the larger population of patients. And of course, when I said that we should actually focus, I mean, definitely we have to do studies focus on single population, but we should actually not forget the rest of the patient. And I just want one thing that could be provocative to everybody. If the immunotherapy fail, are we sure that we are too early abandon all the other aspect of pharmacology that actually could at least, let's say, induce a disease modifying? Glutamate, for example, it's one of them. This is provocative for you.
I just throw out that if this idea of seeding is critical, then there are some really interesting assays now which can detect seeding that could be quite powerful. So I think that's promising.
Gene, did you?
Yeah, I was just gonna. I think these are great points, and I would say, to the point of what is the biomarker you need. I think we've talked about a couple of things that we need to do. We need to identify with greater accuracy patients earlier in the course of their disease. A biomarker for that would be super. We need to be able to, with increasing confidence, know that we're testing the clinical hypothesis with our agent. In the case of anti-synuclein antibodies, we want to know that we've maximally impacted alpha-synuclein. And then, at the end of the day, the hypothesis that you'll assess is whether that has a meaningful impact on the clinical outcome measures that matter to regulators. And I think the third biomarker I'd love to have is one that allows us to get to that last point. Does impacting the biology allow you to have the right impact on clinical outcome in a more expeditious fashion with lower variability such that one can do trials cheaper, easier, and faster? And I think, those are the three sets of biomarkers that one needs. We don't have them today, but I'd say broadly, those are the categories we're talking about up here.
Serena, how worried are you about the lack of biomarkers?
Well, so actually I agree with the rest of the panelists that today we don't have anything that we can hang our head on, and it's a little disappointing. I can imagine a day when we have, for example, an alpha-synuclein PET ligand, how easy my life would be. But of course, by then there'll be other problems. So, it's just a dream at this point. But going back to biomarkers, what their purposes are, and I agree with Fabrizio that we actually really need to be very careful about how we think about biomarkers because, we wanted to be able to use whatever method that we can to identify a patient population that is appropriate for the disease therapy, the modality, the treatment modality. So, genetics is one. But, using, for example, using DAT scan, we've learned a lot over the years, and as also demonstrated in some large data set, like PPMI, that people who have negative DAT scans, they progress at a very different rate. Prior to having a DAT scan, we actually were including all of these patients into clinical trials, not knowing that they are going to be progressing slower. So it actually dilutes out your effect size, and, probably some of the clinical trials in the olden days failed because of that. But now we learned and we can actually apply it. So I think in terms of identifying the right population, there's definitely, I think, a little bit more hope. In the current stage, we actually have a little bit more tools that we can use. In terms of following disease progression and perhaps, demonstrating an effect size of a drug, that I think is a bigger problem. And I have a hard time thinking about what is actually the most promising and most usable, because I'm stuck on Peter's comment about, if you don't see an effect, are you ready to kill a program? Are you confident enough to say, we're not going to pursue this anymore? What if we're wrong? So at this point, I'm not seeing anything that actually can give us that kind of confidence. It doesn't mean that we're going to stop looking. And I think there's actually a lot of very fascinating work in the field that is trying to advance both imaging and fluid. But, I have to say, I can't use them, with any, confidence in the trials yet. But I want to throw out another idea about biomarkers. And we talked a little bit about, clinical trials using clinical rating scale. And we can score them, relatively well, but the scales themselves are very insensitive. So is there a way that we can actually improve on our, the way to measure these clinical symptoms? So I think there actually has been a movement, and it's kind of in vogue right now, that people use wearables. So, at least to me, the idea of using a wearable, there are a lot of problems that we need to solve with a wearable, but the idea is that, when we rate a patient, when they're walking, and on the UPDRS, we actually have a 0 to 4 scale. And we rate, either 0 being normal, 1, 2, 3, 4. But between a 1 and a 2, there's actually a lot of gradation. So can we actually use a wearable and look at this gradation? And can we actually use that to reduce, to this as a more sensitive marker? Maybe perhaps even reduce variability? I don't know. This is something that I think the field is moving into. There's actually a lot of noise around, and I think regulators are actually interested in hearing what we can do in that field as well.
Okay, great. And I will just take this opportunity to mention, since it's come up several times, that the Fox Foundation is offering $2 million to the first group to develop an alpha-synuclein PET tracer. So if anybody here thinks they have one, see me after, and I'll give you your $2 million. So we have a lot of good questions here, and I think what I'd like to do now is just go around to each of the panel members and ask you, what are you most hopeful about when it comes to developing disease-modifying approaches for Parkinson's disease, and what keeps you up at night? So we'll start down at the end. Eliezer, with you.
Well, I think I'm tremendously hopeful. I think all the genetic data that we have, the animal models, the cell-based models, better understanding of the pathophysiology, I think there's a lot of very exciting basic science going on, and I think it's very important to continue to support that basic science and understand again better the relationship between the genetics and the clinical manifestations of the disease. I think that is absolutely fundamental. What keeps me up at night is that, of course, we have only a limited amount of money to do the work that we need to do. And the bottom line is that the talent is there, the knowledge is there, the technology is there. We just need to invest more and have a much greater specific mass to move forward. But those resources are limited. And so where do we put the money? You just were talking about before about biomarkers. Well, if we have a limited amount of money, and we know that the phase 2 trials or phase 3 trials with disease-modifying agents for Parkinson's disease are going to find a roadblock because we don't have a biomarkers, do we just don't invest too much in clinical trials right now and we invest everything we have in biomarkers and in basic science, or do we invest in both of them in parallel but at a lower level? How do we go about choosing investment in an environment where there would be, relatively, there will be a certain amount of money for certain programs. So to me, that is really what it's something that keeps me up at night. But I'm very hopeful that, again, as I said, with the amount of talent that we have there and the technology and the tools, that we'll get to the end of the road and find cures for these diseases, hopefully before 2025.
Peter.
Well, obviously, both of those categories will be my particular target, but I'm really hopeful. We have a compound that gets into the brain really well in animals and should in humans, and we should be able to test our hypothesis in humans. What keeps me up at night is even if we normalize the biochemical defect that's encoded by the risk-associated gene, that may have no effect on the disease because we're normalizing it in a 60-year-old, and that person had the defect since they were born. So that is a really tough problem. I try not to think about it too much, or I try to drink when I'm thinking about it, but it's a really tough problem is how early do these forms like GBA-associated PD, LARC2-associated PD, how early do they really take root? And just by reversing it, you still may be too late. And if it's really, really early, in theory at least, you could imagine a situation where it would be nearly impossible to run a trial. So that's what keeps me up. So.
Fabrizio.
Well, I believe that I believe in immunotherapy, and I believe in immunotherapy because we can start probably later than just at the very, very beginning. Of course, the genetic and the other four, the GBA, that probably is a very interesting target, and we believe in that. I believe that pharmacology can actually really, pharmacology in general, can lead us eventually to slow down disease progression or even cure patients with Parkinson's disease in the future. And the other reason why I believe in immunotherapy, because for the first time nowadays, we are not just looking at the very end of the story. But for example, think about all the interest we have now in microbiota and all the story about the intestine and penetration of the alpha-synuclein getting into the brain and so forth. So we are looking at the general and the patient with Parkinson's disease, trying to understand not only the very end, not only Roma, but actually the pathway, the way going to Rome. And that make me positive in the next future.
Walter.
Well, I'm real positive because I've been around a long time and just seen how good the science is now compared to when I started. So we're learning more and more, and it's just a matter of how much space there is between the rocks that we keep jumping across. I think what keeps me up at night is maybe what was mentioned and what Fabrizio was saying is that, out there, the genetic causes might be 10 to 20%, but there's a lot of stuff out there that's driving Parkinson's, and we have no clue what that is. And that's really what is scary, because we're trying to fight against a process that's been going on for a lifetime, maybe, and it's a lot easier to prevent things than it is to try to stop things once the process starts going.
Serena.
So what's making me hopeful right now is actually seeing the growth of interest in this field. Because, I haven't been around that long, but even in my short period of time in the field, there's actually so much more in the growth in the science. And, if you look at the number of publications, or just look at the attendance of this conference, you see that there's actually a lot of interest out there, a lot of young people who are having, really fabulous new ideas that they're willing to go try it. And a lot of times, that's how we learn and how we discover things. What keeps me up at night? It's hard to say because I don't sleep. No, I'm just kidding. But I think actually is the vast amount of unknown. And we don't know what we don't know. How are we going to deal with them? So like what Walter was saying, we're only probably affecting one small aspect of the disease. So if we fail in this one small aspect, does that mean that all hope is lost? But we don't know what we don't know. So this is actually something that really bothers me, and I hope somebody has an answer for that.
Jim, do you have the answer?
No. Well, I think what makes me most excited is the change in the way we do science now. Whereas, when I started, everybody had their own little laboratory and people put paper on the windows so no one could see what they were doing. Now labs are big open spaces and people share data from the very beginning. And you're much less likely to see a finding coming out of one lab that absolutely cannot be recreated in one way, shape, or form from another lab. So sort of the transdisciplinary approach and data sharing aspects has allowed us really to collect so much data that even when we're not talking about pharmacogenomics, is we have now big data and we can think about things in big ways to answer big questions. I think that keeps me up at night is if we have all the answers, but it was to the wrong question. And so, we're hedging our bets on certain approaches based on what we've done in the past. And maybe what we've done in the past isn't the way that, to find the answer. So I think that's what keeps me up.
And Jean.
Yeah, so obviously incredibly hopeful. I think echoing what everyone has said, I think the sophistication around the science in this space is really just remarkable. It's been great to watch the field grow. I think we have good tools now. We're starting to take good bets in the clinical space, which is incredibly exciting. And the potential hope it offers to the patients that suffer with this terrible disease is incredible. Work that groups like Michael J. Fox Foundation have done with the PPMI initiative and other initiatives to help get us ready for these moments, starting to estimate sample sizes before we got to the trials. I mean, these were well thought efforts that took place before we got these molecules into the clinic, and I think have been incredibly helpful thinking about clinical design. Of course, what keeps you up at night is exactly what was said, the unknown unknowns. We are in new clinical space. We have to select the right patients. We have to select the right outcome measures. And even if we get all of that right, the right biology, the right patients, the right outcome measures, the right clinical studies, the right duration of treatments, we then have to make sure that it's received well by regulatory authorities. We have to make sure that it's received well by payers who, at the end of the day, have to reimburse these medicines, which will no doubt be costly and probably not able to be reimbursed for everyone in the general population. And so thinking about the biomarker efforts, how we further increase our confidence in selecting patients that are likely to convert to Parkinson's disease in a particular time frame, I think will be critically important all the way through the value chain. And we actually have to get every piece of this right at the same time. I think it's doable. I think we've learned lessons from the Alzheimer's disease space, which have been incredibly valuable. And I think those are being applied now. So I'm confident, but I know there's a lot more work to do. And I'm encouraged that there are as many people here today that are willing to fight that good fight with us, and seeing your faces in the audience. So.
Great. Well, thanks to all the panelists. I'll just say that I'm hopeful because we have talented people like all of you working on this problem, and many of the people in the room as well. And the elusive PET tracer keeps me up many a night. So thank you to all of you. And thanks to you for coming.